Yes.
4 zeros. Yes.
Okay, so we wait a minute. Bio on 25. How did I go to and having bio on 25?
Oh yeah, it's complicated.
Today is October 7th. Okay, good. Let me see. Yes, there is a link for the Google Doc master sheet on Moodle. So if you click on that and open in a different tab, that's oh well. Okay, just click directly on it, and it's probably going to pop up. Let me do it again. If I click directly, yeah, it goes to the Google master sheet. So okay, let's see. Okay, who put those things here? Okay, but at least someone is trying to do something here, so it's good. But those are the results. So string 1, 2, 3, 4, 5. A string number is basically a group number, and the OD. Wait a minute. Hold on, that's the OD number? 1.087. That doesn't look right. Someone seems to change the number. Someone changed the number. Don't change the number I put it there. Who wrote it down last time? What's the number I wrote? It should be 2. Is it 2 something? Those numbers are too low to be true. Who wrote it on your notebook?
The one that you wrote yesterday on the board.
Yeah. Someone don't change all those numbers. You didn't do anything about those numbers. That's the number should be.
2.1.
Thank you. Can I have this? Okay. Someone really picked the note. Let me see. There are also 2 strings. I'm going to. So those don't work on this part. If you, I'm not sure what you are trying to do here, and those are your individual ones. And you don't put this in your individual result into entire class. You can work on your own Excel sheet and submit it in your group report, but you are basically overriding the classroom results here. And I'm going to move this away to some other places. Okay, and then go back to the. Don't just the result should be already there. Oh, wait a minute. Sorry, I'm looking at the wrong places. I just realized I'm looking at the spring result. This is the fall. Sorry, no one has. My bad. I'm looking at the spring here. Sorry, this is the okay. I guess I shouldn't just automatically blame someone. Sorry, I apologize. I'm actually looking at the wrong. Okay, sorry. Okay, so this is this is the fall 20. So I basically this is the same document. I can add one sheet, extra sheet. So this is so that's a spring. That's the fall. So look at the fall one, but you can actually look at the spring to see how they do it as a reference. Yeah. So 22. So group 2 seem to have put some result there. So that seems to be too few cell. Oh, I guess I should say that the number of cell per microliter. That's the concentration. So And the same thing at the bottom. And number of cells per microliter at the concentration. And those number, this number is too low to be true, right? Let's just, in fact, you can Google see 1 OD yeast cell, and you can ODs. Oh, that's OD 660, but it's approximate. See, 1 OD, it should be. Where is 1 OD? 1 OD is about 1.85 10 to the 7 cells per, what's the unit here? It didn't say the unit. Per mil. That's per mil. Oh, but we are doing in microliter, so maybe that is the right number. So divided by 3, that would be 10 to the 4. What's the number? So I have 1.85 10 to the per mil. That's 10 to the 4th per microliter. 1.85 microliter. 1.85. Seems to be. Is that correct? 1.71. Look at our number again. Where's our number? Oh, no. Sorry, when I have too many windows, I forgot. Oh, that looks like that. Okay. I don't know. Maybe that is right. But in any case, the top and bottom shouldn't be different by tenfold. They are two strengths. They will be differ subtly. They shouldn't differ by an order of magnitude. That seems to be a. It looks like only one of them should be correct. So okay, but basically that's the master sheet and all the. So on in your own report, you should show your calculation process, but put the final result there in a class master sheet. And in the end, if we plot all this result out and plot the so in the library plot, if we plot all those groups, this is terrible. So we should have if we put the OD at the bottom, OD 600, and then we put the cell number per microliter. We'll have 5 groups. In theory, it should be a straight line. It will be interesting to see what's the final plot going to look like. Yeah, but the point is just it's a learning experience. I'm not. If one group put the point in here, I mean that's the result we got, and you won't be penalized because the point is somehow fall off the line. So, but just yes.
What's your y-axis label?
Number of cells per microliter.
Okay.
Yeah. After there will be at least 2 different measures. We're using hemocytometer. We count the cell concentration, number of cells per microliter. Oh. You want to say something? Oh, okay. And then we can put the OD. We can also put a CFU, colony forming unit there. And there will be, we can also put some other measures. We can put after you measure the mutation rate, it's not really mutation, it's called loss of heterozygosity. You can put the LH frequency there, And then you can measure comes with the cell density. The density plotted, say 10x 10 to. This will be log top. This should be no correlation, but I'm not sure yet. In this experiment, we didn't do any stress factor, so if we have to do an aging experiment, this should increase over time. Or if we put hydrogen peroxide level there, it should also increase sigmoidal shape. But in our experiment, we did only one condition. Okay, so I guess as a group, we should find a way to write a report and finish, put all those values there. I look at the plate, it's actually still, it's going to grow very slowly, yes. You show your procedure how you estimate the cell concentration using the hemocytometer counting data. It's not due yet. It's only the report will be due after we finish all this, but you should start to writing on this.
So
Otherwise,
the first thing that's due is just putting our
right. You need to put the. Otherwise, we won't be able to finish the lab.
One person from each group to put it up there.
Yeah. Someone should put it there. I guess I should put a grade. Put a what?
Or put it up in pieces or all at once.
Your final result should be put in a master sheet. Your individual result keep in your own Excel spreadsheet. How you calculate your result, you should do that in your own Excel. But after you calculate it, put that number there. Making sense? I probably create a say assignment, say 1 or 2 points there. Say once you are done, you as a group you get that point. As a reminder, I mean 1 or 2 points probably a trivial compared to other, but at least it's something as a reminder, I guess. Yeah. Okay, so that's one thing for the Google Doc as a spreadsheet. Okay, going to close this. I look at the assignment. Some of you seem to be prefer to take the second attempt. That's fine. I mean, even for the second attempt, you can still get a 90% percentile. But for the first attempt, with no individual information on which one is correct, still the lowest one is still 78%. So that's still pretty good. So it's that's actually the most challenge. That's the lowest percentile. No, the assignment. The chapter A assignment. Yes. That's a chapter A assignment. Quiz for the quiz. Let me look at the quiz. For the quizzes, oops. There's so many windows I need to. Okay, this is the quiz. For the quiz, let me see. For the quiz, I actually give you a chance to look at it on your own. So let me see. Yeah, so, oops, where did it go? Here. Yeah, self-reflection on exam 1. So those are the part 2 online part. That's part 1, 40-point multiple choice. I even gave you a video tutorial to do the trend line. Those are also the keys to the 2 calculation questions. So, yes, what?
Oh, just saw that we have a guest on Thursday.
Yeah, there's that's because Thursday I will be at the DC for a meeting. So someone else will here give so I'm going to give an assignment. Say you write a paragraph to what this guy talks about. So, yes, what? Thursday, yeah. Yeah, he's supposed to talk about his research on how microbial influence human health. He's from Morehouse Medical School, so, and I never hear him talk about his own work. So you will be the 24 mind here to tell me what he's actually talking about. Yeah. So well. Okay, so that's one thing. Okay, let's look at it. So for chapter A, I look at the chapter 5 chapter. It's similar path. So even though, I mean, I gave all this question. You read about. You think you are doing not to a level you like. It's actually pretty good. Compare, yeah. It's. I'm not sure how other faculty will. I think it's pretty good. So, okay, so okay, chapter 8. Oh, the most difficult question 17. I guess I should tell you what. Not question say if the promoter of a positively controlled operon require activator protein. What's the reason? Because so there'll be a choice. Say A, RNA polymerase can easily recognize consensus sequences. B, they are required to inactivate the repressor protein. C, the promoter have a nucleotide sequences that binds RNA polymerase weakly. And usually that's also not a close match to the consensus sequences. D, they are needed to bind to the allosteric side of DNA polymerase. So D is wrong. B is also wrong. A is just incorrect. So C is the one because in fact we are going to go over the lack operon. That's one of the examples for the for positively controlled operon. That's the C.
So just some clarification. For this week, I wanted to do lecture just on Thursday, and for next week, you will be back for Thursday because you don't have class on Tuesday.
We don't have class on Tuesday?
It's all over.
Oh. Okay, gee. Did I put something on Tuesday there? Okay, sorry. If I well, I won't be here either. So. Okay. I didn't realize Tuesday is also a fall break. Okay, all right. Monday, Tuesday are off next week. So, oh.
We have to reset for the guest lecture that comes on Thursday. We have to decide.
Yeah, you have what this guy talks about.
So,
I think I put his name here. If
yeah,
I yeah. It's a research scientist from Morehouse Medical School and using some genome sequence method to study how microbial influence human health. It's the microbiome project. That's what he told me the topic that he wants to talk about. Yeah. So. This is strange. Wait a minute. I'm trying to do give a slideshow, but somehow the computer crashed. I'm not sure what's the reason. Let me try it again. Do a presentation style. Okay, I'm not sure why. Yes.
are you going to put a link where we upload it? You should put a link where we upload it.
Yeah, that's a good idea. Okay, I'll put a link. You can upload. Yeah. Okay. Yeah. Okay. Oh. Yeah. If you remember this one, we actually mentioned this already somewhat in the previous class. So, but this one, if we where's my laser point? So if the question is, where will be is it true or false? Where is the 5' UTR? Is it in intron or exon? Let me write it down. True or false? 5' UTR is in intron. Is this? Oh, I guess I need to open the Socratic room so you can. Let me do this. I'm going to open a Socratic tool so you can do this. Wait a minute, that's not Socratic.
Yeah, I just have to see.
Okay, this doesn't let me sign in for some reason. Oh, finally, okay. Quick question. So, oh, this will be a true and false. Okay. Confirm. Okay. Yeah, the room name is basically my last name. Zero student answer.
Wait, no, how much you think you want?
Oh, you want to see the PowerPoint? Okay, sorry. Yeah. I'm on operative. Yeah. My room name is basically my last name. Sorry, my full name. I'm going to put this as one of the multiple choice in your next exam. What's the wrong name of the bio 120? You're waiting for a load. Okay. 500. Oh, that's good enough. Or when you are picking 100%, say it's false. So yeah, you got it. Okay. Yeah. So okay. So this is actually a good exercise. What do you think? What's the major group of DNA sequences? Yeah. So that's why I bring this Play-Doh and the toothpick for you. So this will be a group exercise because I don't have too many colors. So you want to first do the DNA, and then you twist it into left hand or right hand.
I don't remember it. To be right, to be left. Maybe it's right. I think it's right from right. Right from right. Yeah. This time you want to put the base pair in it because if you don't put base pair, you will not see the minor group and the major group. The difference. Yeah, you have, so that you want to use a toothpick as the maybe as the base pair. And how many turns per base pair in the DNA type of headache? Okay, then the 2.6 that means 2 turns you should be 5 base pair. Oh, 2.6. 3.6. Oh, that's apathetic. That's protein apathetic. Double check. Someone Google the Wikipedia. 2 point something. I thought.
What you ask?
They somehow that number. Was it 5.3?
What else would it use?
Yeah, for DNA double higher we see in 1 term. For protein apathetic is 3.6 rapidly. Oh. Double headed, you want to know how many per turn.
Yeah, I'm getting.
Okay, well, all right. Okay. All right. So you want to, according to Wikipedia, is that the document? Yeah. Okay, so each group want to build a EMQ. It's either if you lay 2 square as spaghetti and put the toothpick in between them not your base and then you try to twist it to right hand side, and then you will see the major and minor. Okay,
So we're making the right. That's one of the things.
right at the base there. What those? If some of the group, why don't you come and get your favorite color? Oh, you probably need the 2 colors because there are 2 strands. Each group, yeah. Right.
Just a brighter color.
Yellow.
This is that's my It's neon. Oh, but is what do you do? It is like
Yeah.
I think it's the way I think.
I think it's the
Makes it right.
Yeah. You want to If you really want to, okay, if you don't have a need to break the toothpick, if you want to see the menu, you need at least 2 turns. And 2 turns will be 20 of them. Yeah. If you can have 20 turns, right? But you currently 10 basic. Right. Yeah. Besides, if it's basically and stable, you won't be able to. Yeah. Or break it to the small line. Because break it to the small line. If it's large, you won't be able to. Yeah, you want, yeah, it has to be smaller if you want to.
Mayor.
That's not a couple of that's right hand, yeah. Not his right hand. That is right hand. Right hand is always like, I'm looking to see if someone has a water bottle. But that's not the
It's kind of like it would grow back.
Right. So most of the world is basically dominated by right-hand people. So all those knobs, first, yeah, they are all built in right-hand helix style. So in this cap, how do I open? If I twist it with my left helix, it's not. It seems to be tight. If I twist right hand, it's going to open it. Right. So this is the right-hand helix. In fact, all the knob is probably also right hand. If you do it this way, it's going to open. If you do you try to use the left hand, so in fact, if you pay attention to how you adjust the microscope, I know you don't do this, but when I see 20 people try to adjust the microscope, very often your first reaction is to turn it this way. And if it doesn't work, you should remember to turn it backward. You probably don't see this is if the focusing should be turned left, that's actually when you have trouble to focusing, because naturally most people will do it this way. So oh yeah, if you close it, I have a oh, you can, there's some water in the back. If you really want that.
Roll it out after. I'm killing.
That's because it's too long. The toothpick needs to be breaking small one if you want to twist. More narrow one if you want. Yeah.
So you go on the toss of data. I'd rather have lunch.
Okay, so
Those three.
right.
I like going right. That's too cheap. She said,
Oh, now you want to put close together.
Yeah.
Yeah, like, so it's like, they just stick, like, finish, like, sticking, and then, like,
Right.
that, and then just that was in like.
Yeah. Okay, everyone, I have a kind of a working model now. So this will be a major group. You do the major group, that's where the protein will bind. That's just
like, I can't
It's
decide.
because the minor group will be occupied by the base pairs. Like major group is only three. If protein goes to the minor group, you can't break some of the base pairs.
So we got ours.
Ain't no competition, ain't like no real competition, because the real competition.
Oh.
Yeah.
We say like, tell them, hello.
Yeah.
Maybe they should have told me.
I mean, how get that shit going? We have.
Well, protein
We
are one
say,
thing.
Five minutes later. And I start snapping one of the
Well, you may want to take picture of before you do that sample. Yeah. Okay. Yeah.
Maybe I can also.
Instead of I take picture everyone, I think it's better for you to take picture and submit it online. That way save me all the trouble. Why don't you? Yeah. Save and put your group name there. So. Yes.
I mean, I think like that.
Yeah. What's that?
I want to buy
Yeah. No, that's like.
Well, I'm saying,
Yeah.
the turn, the
Right.
base here.
Oh.
Oh, right.
We're taking
Okay, take picture of your art. Probably related online. So you should take a picture probably with the last name of all members. All the participants. Okay.
Let's do the next. Please separate the toothpick from the Play-Doh. Put the. Hold on. You need to take because the next one we still need a Play-Doh. I just realized. Sorry.
I don't know. We need some new Play-Doh. It ain't sticking together.
Okay. I promise after spring break here I will bring a new box of Play-Doh. No, fall break. Why am I always say spring break? Sorry. Sorry. Yeah. Okay. So. Okay, my friend. What is this? I have no idea. Can you tell me what this is? That's like opera? Oh, gee. So what does it do? Hey, it's open book question. You don't have to. Besides, I mean, most of you got it right in the pre-class assignment. So.
You just don't
What? Oh, it's just so much? No, there are just how many items I can even count with my fingers. It should be less than 10 items to go to the shortest line at the grocery store. It's not a long line. So okay. Now I have questions after this, so I want you to write as much information on this. This is also a group assignment, so you can write as much information on this white paper, and then I'll take you to the quiz one. Yeah, the next slide will be a question. But then that's so you also write as much information on the.
So the next question is going to be.
Right. Yeah.
So.
You will need the paper later on for everything. So kind of like factory. Yeah. Right.
My information is what?
Right. It should be, yeah. You could,
Not that.
This is actually
not clear. PowerPoint.
Okay, so I'm going to show the next one. I'll give you some time to this one is also on Moodle, so if you want to look at. In fact, the point is you want to draw the DNA sequence and then use Play-Doh to use some color. So this has to be worked as a group. Use then somehow use Play-Doh make RNA polymerase and then make a repressor, make galactose, and then with certain conditions, I want to see how you put the things around. Yeah. Yes. Okay, you want to draw a light opera, and then draw the repressor, galactose, RNA polymerase, what are the factors there? I mean, and then when I say, then I would say in a condition with low glucose and lactose, how I to put those factors on the.
We don't know.
Yeah, I love fruit. Yeah. Yeah, and enough information for you to answer the question.
It's like
So, but at the minimum, we should draw the up.
lactose.
Oh, wait a minute. Where's my question? Hold on. I made that some question. It doesn't show up. Oh, gee. Hold on. Sorry.
Yeah, they can't.
Oh, this is what I remember.
Yeah, you can't turn it on.
All the feedback.
Gee, I'm sorry. Where did I put my question? Gee. Oh, maybe when it's crashed, I haven't saved it yet. This is Hold on. Oh, here. Okay, finally. Sorry. For a moment, I was really worried. Something would happen. Okay, here's a question. Okay, using so first on the paper you have draw a diagram, and where would the lac repressor be bound in a this is wild type E. coli. I can also open my so this will be multiple choice. Let me see. It's be A, B, C, D. Refresh. Okay, and then I switch back. Yeah. So the question is that E. coli now is growing in a low glucose and high lactose. Where should the lac repressor bind? And so you should draw the sequence and the lac repressor. I mean, whatever you draw it, put it on your lactose process.
Using that.
Yeah, as a one diagram. If you want to do this on the board, first draw that put the repressor there. Do this question on the board.
So draw that shit. You must be not.
Yeah.
Say it out. She won't get up there.
So I will draw. So there will be I. And there's a P for promoter, there's O. There's actually another site called C. I'm going to also put it there. And there's a Z, Y, A. So my repressor will look like.
Yeah.
Okay. Only one person answer. See. Okay. What?
It doesn't show.
Hold on. I'm not sure. Sorry, I'm also new to Socrative, so let's see if I refresh. Three students answer this. Does it work now? Oh, okay. So what? Yeah. So
Yeah, in a while. Why can't you highlight
The last question. It wouldn't be bound.
it won't be found. Where do you put the you say E.
I said if there was no last person, which to the P and O.
If you say like
So
P is proposed.
So-
P is where
so the P is just like the real no, for the when we start.
P is proposed.
But
Okay, let me see your answer.
okay, so I see 50% of you vote for D, and this. Let me see. That is correct. Yeah. So when there's a lot of lactose, why you choose D?
Oh, you said B was the correct answer?
D. D like delta.
Oh, yeah, so. I just chose D.
That's basic instinct. That's something. Okay. Someone else can explain why D is why you choose D.
The lab program basically controls the breakdown of lactose. So if you're in a state of high lactose and low glucose, intuitively you would want the enzyme that breaks down lactose to be expressed. So as a result, you don't want to repress the brown bound. So D would be the correct answer.
Okay. Yeah. That's clear. Thank you. Yay. Okay, so yeah, see, it's you should volunteer for the next question.
Okay, let me change this question a little bit. What if happened E. coli grow in high glucose and low lactose? Hold on, let me copy paste and I'm going to change this question in high glucose and low lactose. Save this. Okay, now E. coli is growing in high glucose and low lactose. And you can, let me, I'm going to refresh the Socratic choice. Wait, it doesn't, I guess I have to open a new question for this to do it. I guess I have to say add another question. Confirm. Hold on. Okay, so the question is again, now E. coli is growing in high glucose and low lactose. Where would the repressor be bound? Yeah.
You didn't add the other one.
I didn't add what?
You said you were gonna add one more element up there.
Oh, yeah, but you don't need this one. There should be a C in front of P. Yeah. For this question,
Didn't it point to that C that's where it's bounded in the other picture that we wrote down? If you go back to it, I can show you.
Which one?
Yeah, it's one of the C.
Yeah, point to the C. What is binding to the C?
RNA polymerase.
No, RNA polymerase bound to the P promoter region. Anyway, that's not what it is. That's explain the block, the blue arrow. And then that's explain this blue arrow. Yeah. Yeah.
For the second one, I like red.
Okay, I have 1 person, 5%. Oh, you all pick B. Okay, I guess it's B. B is correct. Yes. So lac repressor will bind the operator position and block the. Once this is repressor is up here, it basically block this. But what does this? So I'm going to ask you to put actually a repressor at the O places, and then tell me what does it block. Put the repressor at your operator, back to the operator. What do you think that would block? Okay, so what? Put another. So the RNA polymerase actually is pretty big. It's probably one of the largest complex. So where do we think the RNA polymerase is bind? Yeah, you have to bind to the P, and once it's blocked, it's not going to move to the transcription.
Yeah, so it's block transcription. Transcription.
RNA polymerase. So RNA polymerase. Yeah. Now the question is. So I'm going to. That figure is not. Let me see what's the next one. Oh, okay. There's another question. Which part of the DNA region shown in the diagram encodes proteins? Yes. You want to explain? Multiple I'm going to open this again. Confirm. Multiple choice. Yeah, this is a new question. Operator. Yeah. So.
This is question 6.
It's not coming up.
Jamia, you want to volunteer? Explain this one. Which one you pick? Oh, yeah. I'm not asking you. Don't volunteer. You already got your trade. So.
Okay, can I ask you? Oh, that's why Jamia was there.
Yeah.
Now I was too. My bad. I'll let you live.
You can go ahead. She don't know.
You don't know?
Okay. Yeah, I was too. But I don't
You
know.
need to explain why.
What is it? Is it because
So O is for what? O is the operator where the repressor will bind. So O is just a piece of DNA. Right. P, what is P for?
P is the promoter.
Yeah, P is promoter. That's where the DNA fragment on RNA polymerase will bind. So it's also just a DNA fragment. What do, yes.
so it isn't notice or is it?
P is not. It's a promoter. So, well, then there's actually only one choice left, actually. Yeah. So, but you want to explain why C is correct. Yeah.
Where is this at?
In this diagram, the C is not there, but C is where the CRP protein binds. So C is also a fragment of DNA where the CRP, CMP activated protein will bind. That's the C side. That's the CRP binding side. But what is I? What is I?
Initiate. That's the gene that codes for the initiation of transcription, right?
No, try it again.
That terminate the last gene.
No, try it again. Okay, you had tried too many times. And you have let what do you say? It's related. It's getting close. It's getting close. No, what is I?
It's the repressor gene, right?
Yeah, that's correct. Yeah. That's the so I encoded the repressor gene. That's what the repressor is. In fact, I actually draw here.
So I is when the repressor attacks.
Yeah, but yeah. You are. Oh.
Oh, that's what.
Your friend is.
That's how I know it's that's the reason why
Right, those are the structure genes. So that's in the C, ZYA, that's there. Right, the I is also the protein. That's the repressor protein. Yeah. So, I mean, since the P, this is actually quite a stupid question because once P is wrong, A, B, E are removed, so only C are left. It's not, I guess it's not very tricky. It's an easy question. So once you know P is wrong, A, B, E are wrong, C is actually your main one. Okay. Now, here comes a question I'm really proud of. This is a question I completely made up on my own. That's as big as it goes, actually. Because this is PDF image. Yeah, sorry.
Is there a word missing?
There's a word missing? Where?
Something expression.
Maybe there is a word missing. I'm not seeing it. But.
Oh,
It just have to be expression.
this is supposed to be expressed. Or is there a word?
Oh, sorry. Yeah. Sorry.
Oh.
That type of English. Okay, so this is the question. Let me read this question. So the research isolated a mutant in lac operon called lac-X, and this can express the ZYA gene in the absence of lactose. So this is a mutant E. coli, and the lac operon is supposed to express this structure gene when the lactose is around. But in this mutant, even when the lactose are not there, it's still expressed. So that's why it's different from wild type. That's how we call it mutant. Okay, sorry, I'm totally blocking. Okay, so and then in this researchers' lab, there are two more mutant strands, and so those tiny little minus, that means a mutant. Like I minus, that means there is a mutation in like I, which means if the like I had a mutation, that means its product is not going to be functional. The repressor, basically, there's no functional repressor. So that's what like I mutant means. Okay, and like Z is plus is Y, this is functional, like Y plus, like A plus. So that's the genotype, that's the phenotype. And basically this one also expressed like ZYA in the absence of like, because this one doesn't have a functional repressor. So when the repressor are not even there, so it really, the lactose really doesn't matter anymore. So, okay. The second strand, the genotype, in this case, like I is plus. So this one has a functional repressor. But Y is minus. So this one, I forgot which enzyme this one called. The product of like Y is not functional. But in any case, this will induce the expression of LAC-Z, because LAC-Z is the wild type. Okay, so then it actually used something called complementation. So in E. coli, actually in genetics, what complementation is you have two mutants, you put them together, it's wild type, that's called complementation. Or you put two genotypes, you put them together, that's called complementation experiment. Basically, we put two strands, the first strand and your mutant strand, the locus both at the same place. So normally bacteria is haploid. So if I can draw an E. coli cell, and that's, oh, I already have a like. So I'm going to draw a gigantic E. coli cell since I already have this one. So I'm going to wipe my name with kind of misleading. So this is your there's some mutation here. I'm going to call this your mutant lac-X. So this is the nuclear genome, and then with this technique, you can put those a fragment of DNA from different strands also into the same cell, and then the cell will form a partial diploid. That's the way we can do complementation in bacterial cell. So let me put in The second one, I'm going to have a, if I use strain 1, this one will be minus. Those doesn't matter, right? Because those are just those are still considered those are also wild type. This should be sorry, VYA. Those are all wild type, right? So this is basically the first experiment. So we put this. The bacterial cell has both set of genetic makeup here. That's the strain 1 and LAC-X. But the phenotype of these ZYA are still expressed when lactose is not around. That's the phenotype. So all these genes are still expressed when lactose are not around. That's for the first strain. For the second strain, I'm going to use a different color. Strain 2. Strain 2, the I is positive, and those are still their CPO, and the Z is still positive, Y is negative. So when these two together, lactose now is when induce repression. That should be induced expression of lactase. Sorry. Yeah, at the bottom. Yeah. Wait. Lactose. And the second one, induce repression of lactase. No, that should be lactose. Oh yeah, that's correct. Yeah, lactose. No. Release. That's probably a wrong wording. Should lactose now? No, that should be lactase expressed in lactase are expressed only in the presence of lactose. I probably should reword that. Yeah, basically, what? Basically, it's not wild type. When strain 2 plus LAC-X is become wild type. Basically, strain 2 plus LAC-X, and this become the wild type. This is basically the wild type phenotype. So strain 2 and the LAC-X is complement to wild type. This is called complementation experiment. So now, which of the, basically this information, can we tell which part of the mutation is the LAC-X? Where is the mutation in LAC-X is? Let's see. Now, this basically we can use elimination of the wrong one. So the CIP binding site. What does CIP binding site do? That's for catabolite repression. That's because when glucose is around, it really doesn't matter whether lactose is there or not. The lactose operon is always shut off. That's called C is for catabolite repression, and P is promoter. But if promoter is wrong, if the promoter is wrong, no, the gene won't be expressed. So that can be expressed. So it can be promoter. How about LAC-I? Very good. So LAC-I, why that? Can you explain why LAC-I is consistent? Yeah. So if LAC-I is a mutant, it's basically the strain 1, right? Right. So strain 1 has a mutation in LAC-I. Is always express those genes, right? Then if LAC-X also has a mutation here, then the repressor basically doesn't even exist anymore. Right, so lactose is not going to affect how the expression of this will be expressed. So and then, why we put it here and then it become wild type? Because now this one has a functional repressor. It can not only bind this, it's also going to bind here. Right? So this functional copy will bind both operators. So this is a dominant mutation. This will be recessive mutation. This is the dominant locus. Right. So that's what. So once we put a wild type there, it actually binds both promoters. So then it's become a wild type again. Making sense? Yeah.
How would we have gotten to that one though? Like, because it could have been lacti or operator or promoter.
No. What? We just eliminated the promoter, right?
How did we eliminate promoter? It's lacti. How did we eliminate promoter?
Because without promoter, the gene cannot be expressed. The gene was expressed.
Right. Okay. So
Yeah.
operator, it can't be blocked.
That's right.
So
If its operator is wrong, if operator is always wrong, you cannot be rescued. You cannot use this to rescue. Yeah. Yeah. So for your exercise, you can just take picture, submit it online. So that saves me a trouble to take picture everybody. Yeah,
The picture and the video.
yeah. You can when you submit that, you save your put a picture as a last name or something.
You're talking about the DNA.
yeah.
Oh, DNA and it doesn't matter. I just need a name tag. Make sure you are active participant.
Oh,
So, please put the Play-Doh back. I'm going to easy ask for money. So, yeah. I will create a submission. So,
Two questions.
oh, right. Let's see.
I'm iffy on what I got, so of course I know.
I think for the exam part, I'll give it back.
I wasn't here.
Someone probably. Oh wait, no, it's still. All right. Oh, some of you, I realize I still have some of your exams, so probably also we don't have any. You can come and pick them up. Oh, okay.
No, you know what?
Oh, you have the.
I mean, he come to school, so.
Oh.
You were like, I was so ready to what you had to say. Okay, text me.
No.
Okay, well, this is like that eight? That was eight over, I was.